Hi there. In this video, I'll be talking about the cytoskeleton and various cytoskeletal elements, including actins, myosins, microtubules, intermediate filaments, and then cell walls, especially those in plants. If the cytoskeletal elements are all removed from a red blood cell, the blood cell looks like this, so that things will round up or form spheres rather than form the characteristic shape that they normally form when they're active and functional. So this is what they would look like normally. And in order to hold them in that shape, then certain cytoskeletal elements have to be present, including some embedded in the membrane. Actins are involved, and then other cytoskeletal elements, including spectrin and others. This is an artist's depiction of what's going on inside of the cell, a red blood cell. So here's the plasma membrane with glycoproteins coating the outside, as well as other proteins attached to the membrane on the interior surface. And some of those are cytoskeletal elements, including actins would be over here, and spectrin ties those together, and they have embedded in the membrane other proteins called anchors that will anchor those to the membrane so that the membrane can be pulled and pushed in various ways to form the final shape of that cell. Also, in this cell, you can see all of the hemoglobin molecules just packed into that cell. So that's the approximate concentration of those hemoglobin molecules that are picking up the oxygen and carrying those to the cells and tissues. Here's a schematic of how some of those proteins interact to form part of the cytoskeleton. So there's glycophorin, which forms part of the anchor, and anchorin, which also forms part of the anchor. And then there are small snippets of actin molecules through here, which act to attach a different protein, and then spectrin, which forms these bridges between each one of those anchoring units to push the membrane in one direction or another. So here's a more detailed diagram of some of the proteins involved in this cytoskeleton, which pushes these mainly animal cells into their final form. So there's a protein that's just listed as band 3 on a gel, which is a glycoprotein. There are sugar groups on there. There's also another protein called glycophorin, and note the alpha helices going through the membrane. Those are attached to other molecules. This one's attached to anchorin. This is attached to protein 4.1, which hasn't got a name yet. And then actin, and those are all connecting with spectrin, which then acts as the bridge between those anchoring units. In addition to what I just mentioned with actin, actin can form many different structures in cells and perform many different functions. One of the ones is this motor protein, which is in muscle cells, which we've already partially discussed. It starts as monomers. There's a monomer sequestering protein, so there's always plenty of actin around. But there are nucleating proteins which cause the addition of actin in just one direction, and this would be to push out a particular part of the cell membrane. There are severing proteins which will break down the actin. There are cross-linking proteins here that connect with actin, capping and blocking the ends of actin, side branches such as tropomyosin, myosin, and things like that are in muscle cells also. So myosin and actin both are involved in many, many cellular processes in the cytoskeleton of cells. Also, there's one other here in the bundling protein, which makes extensions of the cell membrane that are called filopodia, that is small feet that go out from a eukaryotic cell to then attach. They use other proteins to attach to a surface, and they can then pull the cell towards that direction. So they're essentially small feet that are extended out. And that's all caused by the actin pushing out those regions of the membrane. The addition of new actin monomers and dimers to the growing chain is facilitated by the addition of ATP to the actins. And so those activated actins, which are now phosphorylated, attach to one end of the growing chain, so you can grow out this direction. If they lose the phosphate group, then they're more likely to dissociate. So at this end, with bound ADP, they tend to be loosened up, and then they can be added to the other end by, again, activating them by phosphorylating them. So again, you can see why ATP is so important in a cell. It can act on actins, on microtubules, on tubulin, in various parts of the glycolysis, gluconeogenesis, the TCA cycle, all over the place. And you can see here, it facilitates both assembly or polymerization and depolymerization or disassembly of the actin filaments. Here are just a few examples of how actin filaments cause cellular changes, that is, changes in the form of the cell. This is a gut epithelial cell, so pushing out of the actins here causes an increase in the amount of membrane that is exposed to the lumen of the gut, so that more transfer of sodium and sugars and things like that can be accomplished. Here's one where the cell is just spreading out on a surface, and you can see the different actins, and actually myosin is involved in this too, to push those regions out. These are filopodia here in C, so the filopodia are being pushed out. And so this cell would presumably be moving in this direction. And then this is the contractile ring separating, which will eventually squeeze the cell that's just divided. The chromosomes have divided, and the cell is ready to go through cytokinesis. So a ring of actin filaments forms around that and squeezes the two cells apart for cytokinesis. This is a cell that's been labeled with fluorescent dyes. Different structures have been labeled differently. So the DNA is labeled or is stained actually with DAPI, which stains the DNA blue. The green are microtubules, and then the red are actin filaments. And so you can see the actin filaments stretching the cell out into its ultimate form, although the microtubules in many cases are aiding in that process as well. But microtubules, you can see, maintain the nucleus, so they keep the nucleus in the center of the cell or wherever it should be for that particular cell, and then eventually will form the spindle during mitosis as well. If the microtubules and the actin filaments were disrupted at this point, that is depolymerized, and you can add certain chemicals to do that, the cell would essentially round up, and so it would become a sphere. This happens a lot in cancer cells too. Cancer cells often lose their internal structure and tend to round up. When the direction of growth of the actin filaments has been studied, what is found is in these areas, these sort of stretch mark areas of the cell, the growth is in both directions. So it's sort of pushing those parts of the cell apart and stretching the cell. Once you get to the edge, they're also stretching in mainly one direction to push out this edge of the cell, but also pushing outward, sideways, to stretch out the entire cell, to broaden the cell at that point. Once you get to filopodia, they're all going in one direction to push out those cellular feet, essentially, to start attaching to a surface and then retracting again, thus pulling the cell in that direction. So here's how filopodia works. So there's lamellipodium, which is the initial portion of the filopodium that stretches out, attaches to the surface with other proteins. So that's attaching the membrane by other proteins to the surface of whatever the organism is on or the cell is on. And then you start pushing out. It starts pushing out this section with the actin filaments going in one direction, while contracting this end, which is done primarily with myosin attached to the actin. So that's pulling that end. And then it puts down a foot, another attachment to the surface, and keeps moving in that direction, putting down proteins to attach to the surface and then retracting this end. So there's net movement of the cell in that direction. Very often, actin is coupled with myosin. So this is a myosin molecule with usually as a dimer, and then each of the monomers has a head group, and they're joined together, head to end and then end to head here in opposite directions with those heads, and the heads can attach to actin and then move, so you can get movement of the myosin with respect to the actin filament that it's paired with. And to start off this activity, at least in the muscle, so the muscle is specialized tissue, so we're talking about a cell that has a lot of actin and myosin in it, as well as the sarcoplasmic reticulum, and then the tubule, the lumen of a tubule, which attaches to that. So you get an electrical impulse coming through here from a nerve, and that opens a calcium gate in this T tubule or transverse tubules in those cells. And that calcium that's released then interacts with a second calcium channel in the lumen, in the sarcoplasmic reticulum, and that's filled with calcium also. So that opens a gate and releases a lot of calcium from the sarcoplasmic reticulum, and those are surrounding the actin and myosin bundles. The calcium then can bind to tropomyosin, and so here's the tropomyosin that's surrounding the actin, and that blocks a site on the actin where the head of the myosin can attach. So as you add calcium, that causes the tropomyosin to move off of those sites, and then the myosin heads can attach to the actin filaments, and then the movement between the actin and the myosin, mainly the myosin heads, causes them to move with respect to one another, thus contracting the muscle cell, and thus contracting, if you get millions of these together, contracting the muscle itself. So here's a schematic of that. So when the muscle is relaxed here, you have the thick filament of myosin here with the heads not connected to the actin. And then contraction begins when you release the calcium, which binds to the troponin, and that moves the troponin off of the tropomyosin, so that the heads of the myosin can then contact the actin filament, and then through hydrolysis of ATP, the myosin heads become phosphorylated, then they can move, and then repeated over and over, causes movement of the myosin with respect to the actin, and you get contraction of that cell and contraction of the muscle. So here's a step-by-step diagram of how that's happening. So initially you have attachment of the myosin head to the actin thin filament, you have ATP that then interacts with the myosin head, and it releases from the actin filament. You have hydrolysis of the ATP becoming ADP, and that phosphorylates the myosin head, causing a conformational change, so it's essentially cocked. The head then becomes dephosphorylated, and that causes it to attach to the actin filament. It then loses its ADP, which causes a conformational change in the head, and then you get the power stroke from that, moving the actin with respect to the myosin, or moving the myosin with respect to the actin, whichever way you want to look at it. The next set of cytoskeletal elements, which are slightly larger than the actin filaments, are intermediate filaments, and those include keratins, which produce the internal structure for those epithelial cells, vimentin and related cytoskeletal elements found in connective tissue, muscle cells, and glial cells to push those into their shapes, and neurofilaments in nerve cells, and some of those can be quite long, of course, and then nuclear lamins in the nucleus, and those form the structure for the nucleus itself. They sort of form the interface between the nuclear membrane and the chromosomes. Like actins and myosins, these form characteristic structures. So you can start with a monomer. There's the amino end and carboxyl end, and you get a coiled dimer with the amino ends and carboxyl ends in the same place. You can also get two of those molecules coming together, so you get a tetramer, but they're staggered in how they connect with one another. Those can build up further into tetramers, and then eventually into 8 tetramers coming together to form sort of a rope-like filament that is pretty strong and forms a lot of the structure of cells. Many of the intermediate filaments act sort of like rubber bands in that they're somewhat flexible, so that they allow cells and tissues to stretch and not break. So here, if you have cells here, if you have the intermediate filaments intact and stretch the cells out, they actually provide some force to hold those membranes together. But if you get rid of the intermediate filaments and try to stretch those same cells, they actually will break and rupture. And so it does a lot to help the cells to remain elastic and be able to move without their cell membranes rupturing. Nuclear lamins hold the nucleus together and contact the chromosomes as well. So if you look at the nuclear membrane, and inside of that you have nuclear laminar proteins just inside of the nucleus, and attached to that are parts of the chromosomes. Now, during mitosis, for those organisms where the nuclear membrane breaks down, the nuclear membrane actually doesn't completely break down, so parts of the nuclear membrane are still attached to the lamina, the lamins, and those are still attached to parts of the chromosomes. So when the nuclear membrane starts to reconfigure during telophase, those bits and pieces of membrane eventually fuse together. There's some phosphorylation and dephosphorylation going on, but they're already in contact with the chromosomes too. So it's a way for a really coordinated way to maintain some organization of where the chromosomes are, and then surround those with the membrane that most of it still partially exists in in that area and becomes the nuclear membrane, finally reforming the nuclear membrane. The next major group of cytoskeletal elements are microtubules. In astromitosis, they form around the centrosome primarily during interphase as well as during mitosis, and those form eventually the spindle body. So there's monomers of tubulin in that microtubule organizing center there, and then you get polymerization from there to form long microtubules, polymerized microtubules from those centers. As I showed in the picture of the cell with the fluorescently labeled microtubules and actins, in some places the microtubules and the actins and myosins participate in pushing out areas of the membrane or the cell to become these cilia, including in the gut lumen. So that's the case for the microtubules. They form part of that interaction to extend those regions. And in many cases, when you get things extending out, it's caused by microtubules. And in other cases, more by the actins and myosins, or a combination of both acting together. And again, this is a series of micrographs similar to the other one I showed, where you have this picture showing where the actins and myosins are within this cell, and these are the microtubules. You can see in many cases they coincide with the same areas. These are the Golgi. The Golgi are actually modifying some of those tubulin and actin and myosin monomers to help those become polymerized. And then this is a picture also of the microtubules during a different stage of a slightly different cell, just showing the locations of the Golgi with respect to the microtubules and the actins with respect to the microtubules, showing that there's often a combination of things going on to form the cytoskeleton of those cells. Polymerization of microtubules can start from a filament. The alpha and beta subunits come together, and then eventually you get these essentially tubular straw-like structures of microtubules. Although they are fairly rigid, they can be bent, and so there's some flexibility of the microtubules. They polymerize primarily on one end, called the plus end, and the other end is called the minus end, although they can depolymerize from the plus end as well, and also from the minus end. But it requires GTP to add those on. So GTP has to be added to the alpha and beta tubulins, and then you get addition as an alpha-beta unit to the end, the plus end of the microtubule. As GTP is hydrolyzed into GDP, then you start getting depolymerization on that end. In organisms with astral mitosis, they have a microtubule organizing center in the middle of that aster, with a pair of centrioles that are oriented perpendicularly to one another. And these are microtubule structures, which then from that area, there's a lot of monomers of alpha and beta tubulin in there. And then from each one of those nucleating centers, you get microtubules growing out from those, with the plus end at the far end away from the centrioles. If you look at any cells with very long extensions, such as nerve cells with long axons, much of that is pushed out primarily by microtubules. So to maintain these structures with these very long axons requires the center of those cells to be occupied by microtubules that are pushing out those very long extensions and causing those cells to be very elongated. And in a few cases, these axons are over a foot in length. Not only do these help to form the structure of those very long cells, the axons, but they provide sort of a highway for transport of different vesicles, different chemicals, things like that. So there's an inward transport using some microtubule motor proteins, and there are others that are there's outward transport from the cell body to the axon terminal using different motor proteins that are moving along the microtubules. So there are several types of these, several types of dynein, which only moves towards the minus end, and kinesins, which only move towards the plus end. And you'll notice these ones are dimers and these ones are dimers. And through hydrolysis of ATP, they lift up one foot, move it along the microtubule, and then scoot the other foot up, and then repeat that process over and over and over again, essentially walking down the microtubule. And on the other end of those molecules, they're carrying a cargo, usually a vesicle of some sort, filled with certain chemicals. So again, here, this just shows there are several different types of dyneins, and they'll carry different cargo, and several different kinesins, which will also carry a particular cargo. Now, the cargo can be quite large, so the molecule might be this size, and the vesicle might be 10, 20, 30, 40 times the size of that kinesin or dynein. So they can carry quite large cargoes with them and move fairly fast, fairly rapidly down the microtubule. Here are a couple of depictions of the kinesin in this case. There's the heads on that end and going back to this side where it would carry the cargo on the other end. I guess you'd call these the feet and that the head. Here's the movement of those, and you can actually measure. They move quite fast down the microtubule. And here's a model of that where you have swinging over of one head to attach to the microtubule, and then lifting up and moving the other head to move this molecule down the line along that microtubule. If you look on YouTube, there are several animations, movies of these, and it's a little more instructive than this. So look on YouTube under dyneins or kinesins, and they have these carrying very large vesicles, usually, down the microtubule. This is a scanning electron micrograph showing the cilia on the surface of some epithelial cells in the gut, and you can see that these things actually wave. So you're not only increasing the surface area of that membrane, but you're also able to move these through to collect sugar molecules and other things. So you'd also find these on the outside of some cells, either to help movement or to sift through the liquid environment to find sources for food. So although microtubules are fairly rigid, they also can bend. So in the case of cilia, you can have two different strokes of those cilia. One is the relaxation phase, and then there's a power stroke. So you can actually move the organism or water or whatever you want to move across those membranes by moving these cilia either slowly or fairly rapidly. So if you slice through a cilium or what are called eukaryotic flagella, what you find is this 9+2 arrangement where you have 9 of these doublet microtubules and then 2 central elements, as well as some spoke elements that are not microtubules, but they're microtubule-associated proteins, are called MAPs. And so some of these MAPs are dyneins, nexins. The nexins sort of hold these structures together but allow some flexibility. And then there are inner and outer dynein arms that interact from one microtubule to the next. There are also radial spokes, etc., holding the structure together in the center. And so these are able to move and bend that cilium or flagellum by hydrolyzing ATP, and one end of the, in this case, a dynein is physically attached to the microtubule. That becomes the cargo. And then the other end has those heads that can move with respect to the other microtubule. And so once you cause those heads to move, then you're getting movement of one of the microtubules with respect to the other one. So you can have two types of movement. One is shuttling these things with respect to one another. But if you have them within a membrane structure that is actually held in place by other things, then you get a bending. You still get a movement of one with respect to the other, but what's happening is you get a bending of that structure. And so, for instance, in a sperm or a choanoflagellate, you get this tail moving of that cilium or flagellum. The final cytoskeletal element that I want to talk about are cell walls. Now, in this case, it's for a plant cell, which is made up of cellulose. But fungal cells and other types of cells have chitin walls, not cellulose. And then there are many organisms that have silicaceous cell walls. A lot of the protists, such as dinoflagellates and stromatophiles and things like that, different types of cell walls. But I just want to talk about plant cell walls for the moment because they've been more thoroughly studied. So if you digest away the cell wall by using cellulase on a plant cell, it rounds up. It looks like a sphere. These are called spheroplasts or protoplasts. And they're used in order to get DNA into the cell. So a lot of times this is where genetic engineering of plants starts, by making spheroplasts and then injecting or somehow having the plant take up foreign DNA into those cells. So plants with cell walls look like this, or many look like this, where now you have a rigid cell wall between each of the cells, or containing each of the cells, with the chloroplasts inside. Take the cell wall away and they all become spherical. Primary cell walls look like this for the most part. There's the cell membrane here, the plasma membrane, and then outside of that you have these cellulose microfibrils. Usually in one layer they'll be pointing all in one direction, the next layer they'll be pointing in a different direction, and the next layer they'll be pointing in a different direction. And these are laid down actually from the inside of the cell, which is directing a cellulose synthase complex that's making these fibers of cellulose on the outside of the cell. They also are connected together with a different molecule. These are pectins, as well as hemicellulose. So altogether, these form a network of very strong, tough molecules. There is some flexibility in the cell wall, but not a great deal. Then in between cells, you have what's called the middle lamella, which is an aqueous environment that's then between another cell wall and this cell wall that's shown here. And then again, if this was a fungus, then you wouldn't have cellulose, you would have chitin molecules, chitin filaments that would form a network that would form the cell walls in those cells. Here's an SEM picture, scanning EM picture, showing those layers that are built up in one direction in one layer and in another direction in the other layer. And this makes an incredibly strong, tough cell wall or cell structure, kind of like plywood, where they have one layer where the fibers are always going in one direction, and then the next layer has fibers going in the opposite direction. It makes it so much stronger than if you have the same thickness where everything's going in the same direction. That's actually not as stable at all. As I indicated, these are directed by microtubules. So here's the cell membrane here. And embedded in the membrane are these cellulose synthase protein structures. And there's the microtubule inside the cell, and they're directing these cellulose synthase molecules, part of which are poking out of the cell wall or cell membrane. On this end, you're getting sugar molecules that are being, sugar monomers are being fed into the system here. Those are then producing the polymers, the sugar polymers, which are the cellulose, and they're running along the same direction as the microtubule. So these are actually traversing or moving along the microtubule, and as they're doing that, they're extruding this cellulose material, thus forming those long strands of cellulose all in one direction at a time. So this is cellulose here, these long fibers within the cell wall. And if you look at each one of those fibers, they're made up of glucose monomers. So it's glucose, 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 and then those are bound together with other cellulose molecules to form these thicker fibers. And then you can see in this picture the layer underneath that is going in a different direction, and those are long cellulose fibers as well. So as I indicated, that is producing the primary cell wall. But if you look in wood, these would be wood cells, you have secondary cell walls. So within the outside, which is the primary cell wall, then you start building more cell wall next to that cell wall there. So the cell actually, the cytosol, keeps getting smaller and smaller and smaller as these secondary cell walls are formed to produce the wood. So how much wood is built up and how many cell walls are built up depends on the species of tree or plant. So in this case, you have the middle lamella separating one primary cell wall from another primary cell wall. And then you have secondary cell walls added to that. And in each case, the orientation of the cellulose changes from one layer to the next to the next. And this is very specific to each of the tree species. So by looking at the layers of secondary wood or secondary cell walls being produced, you can tell what species of tree it is or what species of plant it is. Okay, so that's all I wanted to say about the cytoskeleton. Talked a little bit about actins, myosins, and then intermediate filaments, which are a little larger, and then microtubules, which are a little larger than that, and then finally cell walls. Okay, thank you very much. I'll talk to you later.